Thank you. Yes, thank you again for the invitation to speak with you all here today. So I'm going to dive right in and talk about our work on SARS-CoV-2. So as we all know, SARS-CoV-2 is an ongoing pandemic that causes COVID-19. And SARS-CoV-2 is actually a virus, an envelope virus, which targets cells of the lungs and can lead to severe complications, including death. And so if you're looking at the virus as shown here through this cartoon depiction, it's an envelope virus made up of multiple proteins that are exposed on its surface. The most important of these proteins being the spike glycoprotein, shown here in the cyan, purple, and gray colors. This protein is involved in attachment to the host cells and leads to entry of the virus into its target cells. As such, the spike glycoprotein is actually responsible for entry and is also the target of most of the neutralizing antibody responses that occur from adaptive immunity. So if you look at closely what happens when the SARS-CoV-2 spike interacts with the host cell, it has to undergo a conformational change. So in this case, you have the viral membrane here, and you have the host cell membrane here with the host receptor ACE2 shown here. So in order to interact with the host cell, first there has to be a conformation of this receptor-binding domain, which exposes this binding site for ACE2, and that attaches and leads to huge conformational changes, which then allows for the virus membrane to fuse with the host cell membrane, releasing the viral genome into the host cell for replication and further production of new viruses. So if we take a closer look at the spike glycoprotein, what you can see is the first conformational change that it must undergo. And so shown here on the left, you're looking at viral spike protein with its RBDs shown here in red in this closed conformation. But then, in order to interact with the ACE2 receptor, these RBDs have to flip up and expose the ACE2 receptor binding motif here at the tip, which then allows it to interact with these cells. Because of this conformational change and inability to mask this region upon this up conformation, the RBD is actually one of the main target for most of the neutralizing antibody responses. Because if you can block this interaction with ACE2, then obviously you can help neutralize the virus. So just so everyone's on the same footing, antibodies, as we know, are serum proteins that combat pathogens, and they're made up of two chains, the heavy chain shown here in light blue, and then the light chain shown here in dark blue. And because of the ability of antibodies to recombine through these events called VDJ recombination, you can have a very diverse makeup of antibodies circulating within any one individual. But upon infection with a new pathogen, your adaptive immune response will make antibodies that target that specific pathogen, hoping to neutralize it very quickly. So for my talk today, you're going to hear me talk a lot about antibodies, and what you're going to see in my structure figures are these one region here shown as the Fab, or the which makes up the variable chain and the constant chain of the antibody. And so this region is involved in recognizing and interacting with the pathogen that is being targeted by the antibody. So what I want to show you today really quickly is what we've been working on here in Pamela's lab. And so first question we asked at the beginning of the pandemic is that, are there different epitopes being targeted in recovered individuals? And if so, what is this predominant epitope that is being targeted? And so to do this, we use binding assays as well as a technique pioneered by Andrew Ward's lab down at Scripps here in California called negative stain polyclonal epitope mapping, or MPEM. After we did this, we then decided, well, what actually are these structural correlates of neutralization by these potent antibodies? And to do this, we turned to single-particle cryoEM as well as X-ray crystallography to get high-resolution information about antibodies targeting the spike glycoprotein. So first, I'm going to talk briefly about our efforts to characterize convalescent plasma. So as we all know, convalescent plasma has been throughout the news since the beginning of this pandemic as a possible treatment mechanism or treatment therapy for SARS-CoV-2 patients. And the idea behind convalescent plasma, of course, is that you have people that have been infected with SARS-CoV-2 and recovered, and within their plasma or within their blood are antibodies that are capable of neutralizing the virus. So can we utilize that information in that neutralizing plasma to help patients that are still fighting the virus? So our collaborator, at Rockefeller University, so we collaborate with Michelle Newsom's lab at Rockefeller. And the lead on this project, Davide Morbiani, they actually characterized over 150 different convalescent SARS-CoV-2 individuals. And what was striking right away from this study that was published in Nature back in June was that there was a varying degree of convalescent activity. So what you're looking at here in this plot, as well as in this dot plot shown here, are the neutralization titers, 50% neutralization titers of the convalescent individuals by looking at blocking or neutralizing a pseudotype SARS-CoV-2 virus. So what you can see is that, for the most part, you have some activity and you have some patients or individuals that have higher activity compared to some patients that have almost no activity or activity that's undetectable within this assay. So right away, we saw that there is varying degrees of neutralizing activity, and about a third of the individuals have weak or no activity at all. So why is that? What actually epitopes are these antibodies targeting within this individual? And so we decided to focus in on a couple of different individuals that are starred here. So 2 that are very potent neutralizers, and a couple that are medium neutralizers, and then 1 that is weakly neutralizing. So what we decided to do for our work is to isolate, this polyclonal mix of antibodies from these individuals. Then we decided to do binding assays against not only SARS-CoV-2, but other coronaviruses to see if there is cross-reactivity occurring between after SARS-CoV-2 infection with other coronaviruses. Use this polyclonal EM mapping technique to map what is happening within these people's plasma and see which epitopes are being targeted by the antibodies, and then to use high resolution structural information to actually understand the mechanism by which these antibodies are blocking or neutralizing the virus. And so what I'm showing here now is the initial binding data against SARS-CoV-2, as well as SARS and MERS. Not MERS-CoV, it should just be MERS. So what you're looking at here is binding to either S, which is shown here in the blue, or the receptor-binding domain, that one piece at the top of the spike glycoprotein responsible for entry or grabbing onto the ACE2 receptor on the host cell. And so as you can imagine, if you look at, if you're looking at the binding of these polyclonal plasmas to SARS-CoV-2, you have very strong binding across the board, both to the S protein as well as to the receptor binding domain. And then also you see some binding, of course, to SARS-CoV. So SARS-CoV is about 73% identical to sequence-wise to SARS-CoV-2. So we expected to see some cross-reactivity between SARS-CoV-2 and SARS-CoV, in which we do, and some having some also binding to the receptor binding domain. And then we also looked for MERS cross-reactivity. And so while we see hardly any reactivity against the receptor binding domain, which makes sense because MERS uses a different receptor to enter the host cell, we did see some cross-reactivity against the spike glycoprotein itself, suggesting that individuals that have been infected with SARS-CoV-2 are making cross-reactive antibodies that combine both SARS and MERS. We also did this for the common cold strains and showed that there are some cross-reactive antibodies that bind there. But the question becomes, are those cross-reactive antibodies actually neutralizing? And so we're still trying to figure out if we can identify a neutralizing cross-reactive antibody. So from there, I decided to use this EM mapping technique. And so what you do is basically take the Fab products from these antibodies, and you use that to bind to the SARS-CoV-2 antigen. So what you're seeing here are negative stain reconstructions, in which you have the spike glycoprotein by itself, and then in complex with the polyclonal mixture of antibodies from either this patient CoV-21, CoV-57, or CoV-107. So these all represent, different neutralizing activities. So this one had very high neutralizing activity, medium, and then low neutralizing activity. And so what you can see is that, for individuals 21 and 57, we see a predominant epitope being targeted by their polyclonal antibodies. And so in this case for CoV-21, which had the highest neutralizing activity, its predominant epitope that is being targeted is potentially interacting, or we interpret as being interacting with the receptor binding domain at the apex of the spike glycoprotein. And so this would make sense that, right, you have high activity because the majority of antibodies circulating are targeting receptor binding domain for this individual. For individual 57, they had pretty good neutralizing activity, but what was striking is that we observed binding of the epitope outside the receptor binding domain, as its predominant epitope, which suggests potentially that individuals that have antibodies targeting other regions outside the RBD, these other epitopes could be neutralizing. So this was a nice discovery to see antibodies that combine outside the RBD and potentially have the ability to neutralize. And then for C105, the individual that had almost no neutralizing activity within his plasma, we saw no binding or no predominant epitopes within this individual. Now, that doesn't necessarily mean that they don't have neutralizing antibodies. It's just that we weren't able to visualize it within this assay. And just to show you, again, if you look at the ability of these antibodies to neutralize, so what you're looking at here is actually neutralization for CoV-21, 57, 107. So all of the IgGs, can neutralize quite well, but when you cleave them into these Fab products, we see a loss in neutralization. So for this, this suggested to us that part of the neutralizing mechanism is through avidity effects, which allows, the antibody to grab on with both arms and to prevent, SARS infection. So we have evidence, right, that these polyclonal epitopes can neutralize through neutralization assays and the fact that they're targeting regions known to be neutralizing epitopes. But how can we actually know what the mechanism is? So for that, we turn to high resolution techniques, including cryoEM and X-ray crystallography. So our collaborator again, Davide Robiani at Rockefeller University, he was able to isolate into monoclonal antibodies from these individuals. And so he actually isolated at the very beginning, since these were two of the first individuals to enroll in our study, from patient 21 and from patient 107. And if you recall, patient 107 had very low neutralizing activity within his plasma. And we also weren't able to determine any predominant epitope from the polyclonal EM mapping. But Davide was able to isolate a number of monoclonal antibodies from his memory B cells that had very high neutralizing activity, including this BC003, which had a IC50, neutralizing IC50 of around 5 nanograms per mil. So this shows that even though this patient didn't have a high amount of antibodies circulating within his plasma, this individual does have great neutralizing antibodies that work to neutralize the virus. And so we actually focused in on two of these antibodies from this individual, BC005 and C002, which in later slides you'll see become C105 and C102. So both of these antibodies neutralize with around 30 nanograms per mil IC50, which are very potent antibodies. So we first determined the crystal structure of this antibody C1002 attached to its target, which was in this case the RBD. And so this is the crystal structure shown here when you see the Fab of the antibody C102 here in green, and the RBD shown here in gray. And not to go into many details, but we can see the different loops responsible for interacting with the RBD shown here, which makes up this footprint for the epitope. So the epitope now is shown here in green. And what I have outlined is actually the region of ACE2, the epitope that is targeted by ACE2 for entry into the cell. So what you can see from this antibody, part of its neutralizing mechanism is to overlap with the ACE2 binding motif, shown again outlined here in orange, of the RBD. And so one important thing to note about these types of antibodies, or these antibodies C102, is that its epitope, this region that it's targeting, is only accessible when the RBD is in an up conformation, right? So when the RBD is closed down at the apex, it won't actually be able to access its site, much like ACE2 is not able to access the site unless it is in this up conformation. So we were like, okay, well, here's just more details going into the structure. Just one thing to point out is that for the CRH3 loop, it has a very short CRH loop that has a very nice shape complementary to the RBD, and therefore it has no extra room to fit in longer CRH3 loops. And another thing to point out too was the ability of these, can you get escape from C102 or C105 like antibodies? And so recently, there's been reports of a naturally circulating mutation at this position A475 that can reduce binding and reduce neutralization activity of these antibodies. So it's somewhat of a resistant mutation. So you have a 100-fold loss in potency against this one mutation at this position. But all in all, these antibodies are still able to neutralize. And since they're already neutralizing at very potent levels of 3 to 30 nanograms per mil, 100 loss in neutralizing potency still makes them capable of neutralizing within the context of a polyclonal mixture. So from there, we were like, okay, well, we understand that it only can bind to this up RBD conformation. So what does that look like in the context of a virus? So we turned to single particle cryoEM. And so this is just some data showing this in our initial structure, which we were able to refine to 3.4 angstroms overall resolution. And what we see in the structure is that we had, potentially 2 different states that we could tease out from the different populations of particles. One being this antibody now C105, targeting 2 up RBDs, and then a state in which you had all 3 RBDs in this up conformation being bound by the antibody C105. And so again, if we now look at C105 and its shape complementarity is the same as C102, very similar. And again, just to point out that its epitope, parts of its epitope overlap with the ACE2 binding motif. And so the important things to note again are that these CDRH1 and H2 loops and CDRL1 loops, which are encoded by the V gene of the antibody. So this is the parental gene that is common across multiple antibodies. This is what's actually making up most of its epitope. And another thing to point out with these antibodies, which is very uncommon when you think about antibodies in the context of infection, is that a lot of the SARS-CoV-2 antibodies have very few mutations. All right, so typically you will have multiple mutations that are utilized or they're made to help increase the affinity of your antibodies to its particular pathogen. But in the case of SARS-CoV-2 antibodies, we are seeing almost these parental antibodies without any mutations being very potent and having very high affinities. So this makes the case that if we want to improve antibody therapies, there is room to actually improve these antibodies through antibody engineering efforts based off of our structural biology techniques. And so now we have these 2 antibodies, C102 and C105. And what's interesting is that we and others also identified other antibodies that all have the same V gene usage. So what we figured out was that this class of antibodies that utilizes this gene segment called VH3-53 or 3F66, they all have the same binding mode. They all look exactly the same. So what you're seeing here is a superposition of these of our antibodies with antibodies taken from other patients and from other studies. And so it was really nice to see that we have a consensus of this type of targeting of the receptor-binding domain by these class of antibodies is recurring and found throughout many different individuals. And so when you think about why this is important is that, we know there's a class of antibodies that is very potent. And on top of that, these antibodies can be raised in every individual that has been studied so far. So this suggests that vaccine efforts would likely induce these types of antibodies, which is really good. Yeah, I can skip this slide. So one interesting thing about these antibodies, though, is that they utilize a very short CDRH3 loop. Right? So CDRH3 loops are usually the loops that are heavily involved in binding to the assay or to the antigen. And so having a short CDRH3 loop is very uncommon. But as I showed you in the previous slide, the reason for that is because there's hardly any room to have a longer CDRH3 loop. So what happens when you have an antibody, like in this case, C144 or CoV-8-239, isolated from another group that has a longer CDRH loop but utilizes the same parental gene or germline gene? And so in this case, we were like, well, it must bind differently. So how exactly does it bind, seeing that this antibody, in this particular C144, is actually very neutralizing, shown here in the blue curves. And so I saw the cryoEM structure of that to 3.2 angstroms. And sure enough, what you see is that when you have a longer CDRH3 loop in this class of recurring antibodies, it has to shift its epitope or has to shift its ability to target the receptor binding domain. And in doing so, this allows for targeting of the spike glycoprotein over its epitope in both the up conformation as well as the down conformation shown here on the left in the cryoEM maps. And one thing to point out was that, even with differences in CDRH3s, our antibody was slightly different than the other one, isolated from another individual, although it does also shift towards the right. And so what was really cool about this antibody, C144, that we discussed in great length in a recent publication, is that its longer CRH3 loop actually bridges between the different RBDs or adjacent RBDs. So when you think about targeting of an epitope, you generally think that it's an epitope on a single proteomer. But in this case, CDR8 or C144 makes a quaternary epitope in which the CDRH3 actually reaches across and buries these two hydrophobic residues at the tip of its loop into a hydrophobic patch on the adjacent RBD, right? And so in essence, what this does is actually forces the spike to stay closed and a lockdown conformation, which suggests that on top of blocking ACE2 by overlapping with its binding motif, C144 has an additional neutralization mechanism in which it locks the spike down and doesn't allow it to open up and target its other epitopes. So outside of the C144, we've described other structures, different antibodies, all different potencies. They combine multiple conformations in the context of the spike glycoprotein, including up and down RBD conformations with different stoichiometries. And so in a recent publication, we've decided to kind of map out and classify all these different antibodies. What I'm showing here is the 8 different structures that we classified in that work, showing different classes that bind only the up RBD, combine up or down RBD conformations, combine up or down, but doesn't overlap with ACE2 binding site, as well as those that can only bind up that don't overlap with ACE2 binding site that may also show cross-reactivity towards a conserved epitope. So with all these structures, we were able to, in this classification mechanism, or scheme, I should say, we can now look at potential ways for different therapies. And so, of course, monoclonal therapies are now the new push for treatment of SARS-CoV-2. And one way to think about this is that, you want to have a cocktail of therapies. And so having antibodies that bind to different distinct epitopes that don't overlap would be the ideal case. And so what I'm showing here is that, the ideal case scenario would be to use antibodies such as those in class 3, those in class 2, or even class 1 to get additive effects of antibodies, towards neutralizing the virus. So with that, I'll leave it there and just to say, thank you. So we've been working since March, as I was telling others earlier today, to characterize a lot of these antibodies. A huge staff, a big thank you to Anthony West, Claudia Jett, and Beth Huey-Tubman, who have been working with me nonstop throughout this entire process with our small group of folks. Michelle's lab, Nussenswag lab, Davide Robiani, and Julio, and the clinical team led by Mariana, for isolating all the antibodies and the donors, Beamline staff, in the cryoEM Caltech Center, Naima Sharaf and Doug Reese's lab, and most importantly to all the individuals who donated their plasma and their time to all of our studies. So with that, I'll end, and just to point out and note that I too am heading off to my own position. And I will be starting a position at Stanford fall 2021 in a biology program associated with the ChemH Institute. So with that, I'll end and take on any questions.
Thank you, Christopher. That was an incredible study. I mean, it's a really interesting field, which I haven't really encountered too much of in the past. So I encourage people to ask some questions in the group chat. We have some questions here. So there's one from Sarah, who says, 2 great talks today. Thank you both. I may have missed this, but they are curious. Are all the antibodies that you analyzed IgG? Have you looked at any IgAs? And if so, do you see any differences in the antibody specificity or bound epitopes or mutations between IgG and IgAs?
Yeah, so structurally, we have not looked at the IgAs. We have plans to potentially do this using tomography. But our collaborators Rockefeller have isolated IgAs and looked at neutralization of IgAs. And for the most part, they see the same activity as IgGs. And of course, when you have dimeric IgA, you see a slight increase in neutralization activity and potency, which you expect as you now have 4 arms, so you have even stronger the effects on the surface of a virus. Are the epitopes that are targeted? Most likely they're likely the same. And so in this case, we see a lot of class switching occurring. So initial targeting of the RBD and then by IgMs, and then we see a lot of class switching occurring to produce IgGs and IgA. So yeah, so I predict that most of the targets of the epitopes are likely the same. But again, structural biology would tell us that later on.
Right, thank you. I think Natalie just has a quick question. It might be on the same lines. You might have already answered it, but I'm just going to pass over to her.
It was quite similar, so it fitted well here. I was wondering, similarly to Sarah, that you showed patients that had kind of low IgG reactivity, but were outpatients, so obviously recovered well from COVID. And I just wondered if you had any information on, say, if initially they had a stronger IgM response that would have meant they didn't require more refined IgGs.
Right. Yeah. So that study actually that was looking at the plasma neutralizing activity. So that would incorporate IgMs and IgAs as well as IgGs. And then later on, we characterized the IgGs only. But yes, in that study, they looked at the different responses, and it doesn't seem, that IgM plays some role, but the correlation really is with the IgGs. And so if you plot correlation of neutralizing their response to the amount of IgG titers, that really correlates strongly with their neutralizing activity. And so yeah, so I mean, I think it was quite surprising to see this huge range. And so one critique of our study when we went back, at least Davide and Michelle's group went back and looked at hospitalized patients, and what you see even with the hospitalized patients, those that had very severe COVID infection, which require hospitalization, there was hardly any difference in the neutralizing activity between those individuals as well as those that were just outpatient and had very mild symptoms. So what we could say correlated was, the amount of titers and then the amount of titers that were specific to the RBD. And so that was really the biggest correlate of neutralization activity, was how much IgG was actually targeting the RBD, and that would make the most sense.
Thank you.
Okay. So we have a question from Leslie, our first speaker. So great talk, Chris. I may have missed this. Have you looked into glycans in the binding interface and how they may play a role in virus neutralization?
Yeah, so class 3 antibodies, class 3 antibodies tend to interact with the glycan shown here, position 343. And so one idea about the potential of these class 3 antibodies, and so this binds outside the ACE2 binding motif. And so the question becomes exactly how do they neutralize? If you can adopt the up conformation, which we show in our cryoEM structures, like ACE2 can still bind, so how might these neutralize? And so one possibility is that there is a glycan actually adjacent to the RBD on the N-terminal domain, and through some molecular dynamic simulations, it has been shown that these glycans adjacent to the RBD may be responsible for propping up the RBD into this up conformation. And so it's possible that in the context of an IgG binding, that these antibodies are preventing those glycans from interacting and keeping the RBD in its up conformation. So that may contribute to its mechanism. But outside of that, it's very interesting that, yes, you have glycans on the surface of the RBD, as well as across the surface of the spike glycoprotein, but yet they don't really contribute to masking of epitopes like you might see in, say, HIV, where the glycans play a critical role in protecting neutralizing epitopes. You really don't see that here, as in the sense that, the most neutralizing epitope is the RBD, receptor-binding domain or motif, and that has no RBDs or glycans whatsoever around it, and that is exposed and allows for very potent antibodies to be raised. So SARS-CoV-2 doesn't utilize glycans in the sense for protection as we might think, outside the fusion peptide region, where there are 2 glycans that really protect the fusion peptide, because that's probably the most conserved area of all coronaviruses.
Thank you. So we have a question from Antoinette, who said, great talks. So for Christopher, how would you expand your findings to cover the known strains of SARS-CoV-2 now, and also the ones that we may encounter with any next wave in a predictive sense?
Yeah, so that's a great question, and we are actively working to do that. And I think a number of groups are looking at this. And so one interesting thing about these class 4 antibodies shown here is that they target epitope that is very cryptic, or is cryptic and is conserved. And so in the context of a trimer, you know, this region is usually buried and is only exposed when you're in the up conformation, but it also requires multiple RBDs to be up to really get antibody down to target it. But what is interesting is that, you know, sequence conservation in this region is very high among different SARS coronaviruses, including some zoonotic coronaviruses that have yet to, you know, make the jump into, you know, human circulation. So a lot of focus right now is being made on identifying antibodies that target this region that are potently neutralizing, and could those antibodies then be utilized as a, you know, as a therapy, or can we focus vaccine efforts to raise antibodies that target this particular region, to make it accessible in, you know, in the conformation that will allow binding neutralization at pretty high potency. So yeah, we're actively looking at that, and I think other groups as well, and to have reagent ready for future outbreaks.